We're going to continue on with biomolecules. Biomolecules are also called organic molecules, and they all contain carbon and also hydrogen. There's 4 different types of biomolecules. We'll talk about carbohydrates, lipids, proteins, and nucleic acids. All biomolecules have a carbon skeleton, and then attached to that they have different functional groups which give them their chemical properties. You do not need to memorize these functional groups. So let's start with our first biomolecule, carbohydrates. You can recognize a carbohydrate because it has a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. So for example, glucose has a chemical formula of C6H12O6. Carbohydrates are polar molecules, so they're going to be held together by polar covalent bonds. So they're going to be sharing electrons between the atoms unequally. So that means there's going to be some partial charges, and that's going to make carbohydrates hydrophilic. And because they're hydrophilic, that means that they like water and they will mix with it very well. And that's easy to remember because a great example of a carbohydrate would be sugar. And if you remember, if you put sugar in water, it always dissolves and mixes really well with water. So that's an easy way to remember that. All right, so we have a couple different types of carbohydrates. So the monosaccharide, that is just one sugar unit, mono meaning one. So that would be one sugar. And then a disaccharide, di meaning two. So disaccharide would be 2 monosaccharides joined together. And then poly, meaning many, polysaccharides are going to be 3 or more monosaccharides joined together. Okay, and then it's also important to know what we use carbohydrates for in our body. So we're going to use carbohydrates as an energy source. And also carbohydrates make up part of our cell membranes as well. So we'll look at that in another video. All right, so here are some examples of monosaccharides. So they're just one sugar unit. Probably the most famous is glucose. And when you put monosaccharides into water, like in our cells, they form these ring structures. Some other examples of monosaccharides would be fructose, that's the sugar found in fruits, and then galactose, which is one of the monosaccharides that's in part of the sugar that's in milk. And then here's some disaccharides. So notice we've got 2 monosaccharides. So here's one and here's the other, and they're covalently joined together. There's a covalent bond in between. And on top we've got sucrose, which is table sugar. And on the bottom we've got lactose, which is the the sugar found in milk. And then here's a polysaccharide. So this is glycogen. And notice that we've got lots of glucoses all joined together by these covalent bonds. And so this would be a polysaccharide. So glycogen is a very large molecule. The function of glycogen in our bodies. We're going to be talking a lot about glycogen later. And glycogen is the storage form of glucose in our bodies. So this is how we store glucose. And we're actually going to store this glycogen in our skeletal muscle and liver. And when we're running low on glucose, we can just take our glycogen and we can actually just chop off these glucoses and then release that and then use that as an energy source. So glycogen is really important in the body, and we'll talk more about that later on in another chapter. Okay, our next biomolecule, these are lipids. And you can recognize lipids because they're made mostly of hydrogen and carbon. And the covalent bonds that hold together carbon and hydrogen atoms, they're going to be sharing the electrons equally. So they're going to form these nonpolar covalent bonds. So lipids are nonpolar molecules. So there's not going to be any charges anywhere. So that's going to make lipids hydrophobic. So they do not like to mix with water. And we all know that because if you, for example, take some olive oil and pour that into a glass of water, the olive oil is not going to mix with the water. It's going to stay by itself and will not mix at all. Some of our lipids are amphipathic, so that is a good word to know. Amphipathic, that means that one end or one region of the molecule is hydrophilic and another part is hydrophobic. So amphi meaning both, and pathic means to feel, so it just literally means it feels both ways. So the molecule has both a hydrophilic part and a hydrophobic part. Okay, and so we'll see an example of that in a little bit. Okay, so there's four different classes of lipids that are important in the human body. We've got triglycerides, also called fats, phospholipids, eicosanoids, which are probably new to you, and then also steroids. All right, let's take a look at triglycerides or fats, how they got their name. Tri, meaning three, is talking about these three fatty acid chains that are kind of in this gold highlighted area. Fatty acid chains are really long, lots of carbons and hydrogens there. And they're going to be attached to a glycerol molecule. That's where the glyceride part comes from. Glycerol is just kind of a modified sugar. Anytime you see glyc, think of glucose, okay? So that's just kind of a modified sugar that these three fatty acid chains are attached to. So that is a fat molecule or triglyceride. What we're using fat or triglyceride for in our bodies, it's definitely an energy source. All of these covalent bonds that are joining the carbons and hydrogens together, when we break those release energy. So there's lots of covalent bonds there, and so lots of energy that's stored in fat. And so you can see why the body decided to store extra energy as fat, because we can get a lot of energy out of that when we break down the fat. Fats also are a good insulator, helps keep us warm, and then they also serve as a nice cushion. So if you're sitting down right now, you are sitting on some fat. Okay, so looking at the triglyceride, kind of taking it apart, looking at those fatty acid chains. There are different types of fatty acids. We have saturated and unsaturated fatty acids. Saturated fatty acids, what we're talking about there is they are saturated with hydrogens. And that means you will not find any double bonds anywhere in that fatty acid chain. Okay, so there's just going to be single covalent bonds everywhere. Okay, so that's a saturated fatty acid. Okay, so saturated fatty acids will have no double bonds. And where we find saturated fatty acids, these are generally going to be from animal sources. So like, if you have like a piece of meat and there's some fat on it, that's going to be saturated fat. Or butter that has saturated fat on it, and it's going to be generally going to be a solid at room temperature. There is some plant sources of saturated fatty acids, coconut oil and palm oil. Those are saturated fats as well. The saturated fats are not so good for you. Okay, because what they do is they will raise your bad cholesterol, which we're going to talk about later. And the bad cholesterol, we're going to talk about how this clogs your arteries later, but it will clog your arteries and that can lead to a heart attack or a stroke. So you don't want to be eating too much of the saturated fatty acids. A little bit's okay, but you don't want to have a lot of that. Okay. The unsaturated fatty acids are much more healthier for you. Okay, so they are not saturated with hydrogens, and we do have some double bonds. Okay, like you can see here, we've got a double bond between those carbons there. So that would be an unsaturated fatty acid. So we do have double bonds with the unsaturated fatty acids. Okay, so when you're reading your nutrition labels on your food, a lot of times they'll say, oh, there's monounsaturated fatty acids and polyunsaturated fatty acids. Well, the difference between those two, the monounsaturated fatty acids just have one double bond in the fatty acid, and then the polyunsaturated fatty acid have two or more double bonds. So that's the only difference between them. And both of these are much healthier to eat than the saturated fatty acids, okay, because they're more likely to raise your good cholesterol, and so they're not as likely to clog your arteries and give you the heart attack and the stroke. The unsaturated fatty acids are going to be a liquid at room temperature, and they're going to be coming more from the vegetable sources, and fish is where we're going to be getting these in our diets. Okay. An exception to this rule are the trans fatty acids. Okay, these are unsaturated fats. Okay, and I've got a picture over here. So there is a double bond, but what's happened is that the hydrogens are across from each other. Trans means across. Okay, and notice on this other unsaturated fatty acids, the hydrogens are on the same side. Okay, so this is what makes it a trans fat. Right, so when the hydrogens are on the opposite side. So how'd that happen? Well, this happens when you process your food. So when you're reading your nutrition label, if it says partially hydrogenated oil on it, what they've done is they've taken some unsaturated fatty acids, maybe some vegetable oil, which is cheap, and they've bombarded it with a bunch of hydrogens to try to turn it into a saturated fat to make it solid. And in the process, they've made some of these trans fats, okay, which is bad for you. All right. So these trans fatty acids are the worst ones you can eat. Okay. So they will raise your bad cholesterol the most and will really raise your risk for clogging your arteries and really increase your risk for heart attack and stroke the most. So you really don't want to be eating these trans fatty acids. Okay, so if you see hydrogenated or partially hydrogenated oil on the label, probably don't want to eat it. Okay, and we see that on processed foods. Okay, is mostly where these are coming from. All right. Okay, so so the trans fats, those are the worst. And then the second worst would be the saturated. Okay. All right, another lipid, the phospholipids. These are amphipathic, just meaning that they've got both the hydrophilic part and a hydrophobic part on the same molecule. So if you look at the structure of a phospholipid, remember that the phospholipids have a head region, so that part is hydrophilic, and then they've got two fatty acid tails hanging off the end here, and that's the hydrophobic part. So that's why they're amphipathic. So they have both the hydrophilic part and a hydrophobic part on the same molecule. And remember that the phospholipids are really important because they make up the cell membrane or the plasma membrane. All right, so when you throw these phospholipids into water, like in our body, because they're amphipathic, the phospholipid heads will face outwards towards the extracellular fluid outside the cell, and then also to the intracellular fluid inside the cell. And the fatty acid tails will face to the inside, they'll kind of huddle together. And so they'll form this phospholipid bilayer that will make up the plasma membrane or cell membrane. Okay. And then the other important structure that phospholipids will form is this little round ball that's called a micelle. Okay, and that is just one layer of phospholipids. Okay, whereas a cell membrane is made up of 2 layers of phospholipids, a micelle is just one layer of phospholipids. Okay, and these micelles actually will form in your intestines from the lipids that you eat in your diet. Okay, so we will see these again when we get to the digestive system. Okay, another lipid. These are called eicosanoids. So these are probably new for you. These are actually made from the phospholipids in the cell membrane. And what happens is the fatty acid tails, they get converted to this arachidonic acid, and then that gets converted into these eicosanoids. The name is really funny. Eicos, that prefix means 20, and it's just referring to the fact that if you count the number of carbons up here, there are 20 carbons, and that's how they named this. Okay. There's lots of different types of eicosanoids. Eicosanoids is just kind of a general name. Some of the different types of eicosanoids that we're going to learn about later, there are prostaglandins. Prostaglandins, there's a lot of different prostaglandins in the body. Some of them are involved in inflammation. Others are involved in, like in women, menstrual cramps. You can blame prostaglandins for that. They actually cause the uterus to contract and cause that cramping, so that's great. Eicosanoids will also form thromboxanes, and you might have heard of a thrombus. Thrombus is a blood clot. So thromboxanes are involved in clotting the blood. And we also oftentimes don't want the blood to clot. So if there's no cut, we want to be releasing these prostacyclins, which are eicosanoids that prevent the blood from clotting when you don't want it to clot. Okay, so we'll talk about those later. And then another type of eicosanoid are called leukotrienes. So these are produced by some of our white blood cells, and these leukotrienes are involved in inflammation. Okay, so we'll come back and talk about these eicosanoids in another chapter. Okay, so just you've heard about them right now, and so they won't, be new when you hear about them later. Okay, and then our last lipid are steroids. So steroids have a very recognizable structure. So they're kind of shaped like a plate, and they have these 4 carbon rings that are all joined together. And cholesterol is part of the cell membrane, so it's inserted into our cell membrane there. And if you remember its function in our cell membrane, it's there to help maintain the correct fluidity of the cell membrane. Because you don't want the membrane to be too solid or too runny. And so the cholesterol will make it just the right consistency. So it helps with that. And then other things we use cholesterol for in our bodies, because I know cholesterol kind of gets a bad rap, but we actually do need it. So besides using it as part of our cell membrane, our liver will also use it to make bile salts, which we secrete into our small intestine, and that will help kind of break the lipids in our diet into smaller chunks so we can digest it easier. And then we also take cholesterol and form that into our steroid hormones. And I've got listed there a lot of our steroid hormones there that we're going to be talking about. So all of the sex hormones, estrogen, progesterone, testosterone, stress hormone, cortisol, and we'll talk about aldosterone later, and vitamin D3. Those are all made from cholesterol. Okay, proteins. Proteins are polymers of amino acids. Amino acids are the monomer, the little building block, and we're just going to join those together to make our protein. So an amino acid, pretty easy to recognize. They have this central carbon, and attached to that they've got an amino group, so that's where amino acid came from. And then on the other end they've got this carboxyl group. And the carboxyl group is actually acidic. It actually releases hydrogen ions, and so that's where the acid part came from. And then what makes all the amino acids different from each other is this R group. Okay, so that's different for the different 20 amino acids. Right, and then the type of covalent bond that's joining the amino acids together to make the protein, these are peptide bonds. And the proteins have a lot of different functions in the cells in the body because they have lots of different shapes. So some of them are receptors, some of them are carrier proteins used to transport things across the cell membrane. Others act as enzymes. Some of them are hormones like insulin. Others are contractile proteins like in our muscles. Some of them are used for defense like antibodies. And others are used for structure, okay, like collagen, elastin in our skin. We've got the keratin making up our nails and hair. And then we've got different levels of protein structure, different levels of folding. So let's go through that really quick. So remember the primary protein structure, that's just the linear sequence of the amino acids, so just what order the amino acids are in. And remember that's coded for by the DNA. So you've got your gene coding for a protein, and the DNA will tell you what sequence of amino acids to put in. Okay, and then the secondary protein structure, we're just going to take that primary structure and then start folding that. And then we get the secondary protein structure, which could either be an alpha helix or a beta pleated sheet. So the alpha helix, that one we just have kind of this coil or spring shaped. And then the beta pleated sheet, it looks like an accordion shape to me. Okay. And these are all going to be held together by hydrogen bonds between the amino acids. Okay, and then we'll just take that secondary protein structure and fold it up even more. And then we get this tertiary protein structure. So this is just the overall 3D shape here of this single polypeptide chain. That's your tertiary protein structure. Okay, and this is all going to be held together by interactions between the R groups on the amino acids. Okay, and in the quaternary protein structure, you have 2 or more polypeptide chains that are folded together and all held together. So like this is a hemoglobin protein. It consists of actually 4 polypeptide chains that are all held together, and that makes the hemoglobin protein. So that'll have the quaternary structure. Okay, and then this is just a nice summary of the 4 levels of the protein structure. And remember that the shape of the protein determines its function. So we've heard that before, right? Shape determines function, or structure determines function. Okay, and a lot of our proteins will be 2 basic shapes, either fibrous or globular. The fibrous proteins are shaped like a rope, so they're going to be used for structural things, so like collagen, that's used to strengthen the skin. And then the globular proteins are going to be round, okay? And they're not used for structure, they're used for all other kinds of jobs. So like insulin would be a hormone used to communicate things in the body. And then like this myosin protein here, this one's kind of a mixture. I think it looks like a golf club to me. So you can see it's got a fibrous tail and then this globular head. So I guess it couldn't decide which one to be, so it decided to be both. And then our last type of biomolecule are the nucleotides and nucleic acids. So the nucleotide, that's the monomer or the building block. So remember the nucleotides, here's a picture of a nucleotide over here. It has 3 components. You've got a 5-carbon sugar, which will either be deoxyribose if you have DNA or ribose if you have RNA. And then there's the nitrogenous base, which will either be an A, T, C, G, or U. And then you've got your phosphate group. Okay, so that makes up your nucleotide. And then we've got ATP is actually an RNA nucleotide that's used to transfer energy. And then also we've got some other ones that I know you've seen before. We really won't get into these, but the NAD plus and FAD, those are used to transfer electrons like in the electron transport chain. Cyclic AMP and cyclic GMP, we'll talk about those later. Those are used in cell signaling pathways. And then putting the nucleotides together to make a polymer, that gives you your nucleic acid, your DNA, and RNA. So, okay, and then here's just an example looking at nucleotides, looking at ATP over here on the far right. Has 3 phosphate groups, ADP only has 2 phosphate groups, and then AMP just has 1 phosphate group. And the one that contains the most energy would be ATP, right? because the energy is stored in these covalent bonds joining these phosphate groups together. So the more phosphate groups you have, the more energy that's stored. So ATP would have the most amount of energy out of all three of these. Okay, and then here's just cyclic AMP. It's just AMP, but how it changed is they just took the phosphate group and they just made a circle out of it. Okay, and we'll talk about this one a little bit later, but it's used in the cell signaling pathways, so for communication. Okay, and then just remember the difference between DNA and RNA. DNA stores the genetic material. You've got 2 strands, double-stranded. You've got T's instead of U's. And then you've got deoxyribose as your sugar instead of ribose. Okay, and RNA is just a single strand, and you've got U's instead of T's, and you're using ribose as your sugar instead of deoxyribose. So